Okay, hi, I'm Banu, and I will present TargetCall, our work, which was an ETA study by the software research group. Our motivation in this work is that basecalling consumes up to 84% of the total execution time in nanopore sequence analysis, and it becomes a major computational overhead for the pipeline. We observe that much of this computation is wasted, as many reads do not match a reference genome of interest, and they are discarded since they are useless for the target genome analysis. Our goal in this work is to eliminate this wasted computation in basecalling while maintaining high accuracy, scalability, and adaptability. For this, we have the key idea of applying a pre-basecalling filter that would filter out the useless reads before the basecalling step with high accuracy and performance. We propose a target call for this filtering step, which works in two main steps. First, light call, which is a lightweight basecaller, computes the noise reads with high performance that are highly accurate but noisy compared to the state-of-the-art basecallers. And the second step is similarity check, which compares the noise reads to the reference genome to determine if the read is useful. Our evaluation shows that target call can provide 3.3x speed up over the state-of-the-art basecaller used in the basecalling step by filtering out majority of the user reads with high sensitivity. And TargetCall achieve better performance, throughput, recall, and precision than prior state-of-the-art target sequencing approaches when they are repurposed as pre-basecalling filters. I will start with background and motivation. The genome sequence analysis enables us to determine the content of a DNA sequence of an organism, and it's important in many areas, such as in precision medicine, outbreak tracing, and understanding of evolution. Nanopore sequencing is one such technology which is widely used, and it can provide long reads of tens of kilobases per length with high throughput and low cost. And the traditional nanopore sequence analysis pipeline starts with genome sequencing, where the DNA fragments are converted to raw signals when they are passing through a pore. Then these raw signals are converted to a sequence of bases called as reads during the basecalling step. The next step is to match these reads to a reference genome to predict if the read is going to be mapped, meaning matched to the reference genome, or if it's going to be unmapped. And this stage is called as read mapping stage. Prior studies shows that basecalling consumes up to 84.2% of the total execution time, while the remaining downstream analysis takes less than 16%. And it concludes that basecalling is a major computational overhead for the traditional nanopore sequence analysis pipeline. Our observation in this work is that what worsens this situation is majority of this computation is wasted because majority of the basecall reads are discarded after basecalling step during the downstream analysis. One such application is SARS-CoV-2 genome assembly, where less than 1% of the reads are useful, even though the entire dataset is processed fully during the basecalling step. And the typical reason why 99% of the reads are useless is that they do not match the reference genome that is targeted by the specific application. That's why we conclude that useless reads cause significant waste of basecalling computation in traditional nanopore sequence analysis pipeline. There is one alternative to this, which is called targeted sequencing. And in traditional pipeline, we sequence all the reads and basecall all of them. Instead, what we do in targeted sequencing is that we selectively sequence only the useful reads. This is enabled by a functionality of the nanopore devices, where the device can stop sequencing a certain read with a reject command. And in targeted sequencing, the rejected DNA fragments are removed from the pore, so they are not fully sequenced, and then the accepted ones are fully sequenced and sent to the basecalling step for further analysis. But this targeted sequencing requires a method to identify which read is useful for the downstream analysis. Our analysis shows that the current methods to identify which reads are useful for the targeted sequencing approaches suffer from at least one of the three following key shortcomings. First, they have low sensitivity and they falsely reject many useful reads that should have been sequenced. And second, they have poor scalability in the sense that their performance and accuracy in identifying useful reads drop significantly when the reference genome size increases. Or they have lack of adaptability because they use neural network classifiers to classify if the read is going to be useful, and these networks need to be retrained whenever you change the reference genome or the read set that is targeted by the application. That's why they're not easily adaptable to different use cases without costly neural network training. And that's why we conclude that these approaches cannot be a robust solution for eliminating the wasted computation in base calling. And instead, our goal is to eliminate the wasted computation in base calling, but while maintaining high sensitivity in keeping useful reads, being scalable to large reference genomes, and maintaining adaptability to different use cases. And we propose TargetCall with a pre-basecalling filter for this purpose. But before explaining what is pre-basecalling filtering, I will remind you shortly what were the options. So the first option was a traditional pipeline where we sequence and base call everything. And second option is to do targeted sequencing and sequence reads selectively and then do base calling everything that is sequenced fully by the pipeline. What we propose instead is pre-basecalling filter, which is selectively base calling useful reads. So it's different than the traditional pipeline because we do not base call everything. And it's also different than the targeted sequencing pipeline because we do sequencing on entire data set and then we do filtering before the base calling step. This kind of enables us something because we do the filtering after the sequencing is fully performed. So we have the raw signal of the entire read when we are trying to classify the read as useful and useless. We don't have this opportunity in targeted sequencing because we have to predict if the read is going to be useful while the read is still being sequenced. So we have much more information in this formulation of the problem. And how we do it is we have a new step between the sequencing and base calling step, which is called as the pre-basecalling filter in our paper. And this filter needs to identify which read is useful and useless, again, using the entire raw signal information because we do this step after sequencing is fully performed. And anything that is identified as useless by this filter is dropped from further analysis, and then the raw signals that are useful for the further downstream analysis is forwarded to the base calling step. So the high-level goal of TargetCall is really to just predict if the read is going to be useful for the downstream analysis. And it does so using two steps. The first step is to do some light base calling, and we call the component as light call, which performs noisy base calling in a very fast way compared to the state-of-the-art base callers. And the second step is to do a similarity check, which would compare this noise reads to the reference genome to predict if these two things are going to be similar, and then if the read is going to be useful based on this similarity score. And if the read is useless after this analysis, the analysis of that read is stopped at this point, and the reads that are identified as useful at this step is forwarded to the base caller so that we maintain the base calling accuracy for the useful reads. And TargetCall has high sensitivity because its similarity check component is highly accurate. And it has good scalability in the sense that it scales well to the performance and accuracy scales well to the reference genomes that have longer length. And it's because the model of light call, the performance of the model of light call is going to be is independent of the reference genome size. And finally, it's adaptable because we do not need to retrain anything again for different use cases because we designed light call to be generic, the model of light call to be generic across use cases, unlike the prior tools. And that's why we say that TargetCall can provide a robust solution for eliminating the wasted computation in base calling. Before evaluating how it can be useful, we just show the two components of TargetCall in a bit more detail. The first component is light call, which is a lightweight base caller that performs noisy base calling compared to the state-of-the-art base callers. And light call is designed based on simplifying the state-of-the-art base callers model architecture. The simplification has happened in a way that the performance is improved significantly, but most of the accuracy benefits of this complex neural network model from the state-of-the-art base callers is preserved. And we do so like this simplification in 3 steps. First, we reduce the channel sizes of convolution layers. And then we remove skip connections that are present in the state-of-the-art base callers. And finally, we reduce the number of basic convolutional blocks compared to the state-of-the-art base callers. And the final large call architecture is composed of repeated convolutional blocks, where each block has 1 or 2 units that include group convolution, point-wise convolution, batch normalization, and ReLU layers. The second component of TargetCall is similarity check, and it gets input from the previous step, which is light call. The input is noisy read coming from it. And the second input is the reference genome, which is determined based on the application we have. And the goal is to predict if these two things are similar to each other. And depending on the answer, it labels read as useful and useless. And then this labeling is dependent on the application and again. And this problem is very similar to the read mapping problem. That's why we use the state-of-the-art read mapper Minimap2 for this module. And we show that if we use similarity check, which is like Minimap2, on the reads that are coming from the light call, so on the reads that are base called in a noisy way, we can still achieve very high sensitivity in keeping useful reads. So Minimap2 can tolerate the erroneous bases coming in the noisy read. And we further show that this 0.5% loss of the useful reads can be tolerated because we have high sequencing depth of coverage in our data set. And before going through the evaluations, we can check what kind of use cases we can have for TargetCall. We show different use cases. We actually show 3 use cases to show how scalable and adaptable TargetCall is to different use cases and different reference genome sizes. The first use case is to determine or find or filter reads coming from a small reference genome, which is SARS-CoV-2 in this use case. And in our reads, we have the SARS-CoV-2 and human ones, and we try to identify the ones that are coming from the SARS-CoV-2 reference genome. So in this use case, if the read is similar to our reference genome, it's identified as useful. And the second use case includes a bit more complex reference genome and reads to show that TargetCall can actually work when the reference genome has multiple viral strains and the read set has multiple viral reads and multiple bacterial reads. And again, anything that is similar to the reference genome is identified as useful in this use case. The final use case is sepsis detection, which is to show TargetCall can work very well when the reference genome size is large. And here the goal of use case is to just filter or determine any bacterial reads that is coming from that is in the sample. And here we don't know which bacterial reads we have in the sample. That's why we just determine the human ones and delete them from our sample. And that's why in this use case, if the read is similar to the reference genome we have, which is a large human reference genome, the read is identified as useless because the goal of this use case is to identify the bacterial reads in the sample. We have 2 sets of experiments to evaluate TargetCall, and the first one is to show the benefits of pre-basecalling filtering in general. For that, we compared Bonito, which is a state-of-the-art base caller developed by ONT, and Bonito preceded by TargetCall to filter out users' reads before the base calling step. And then our second set of experiment is to compare TargetCall against these targeted sequencing approaches. We actually use only 2 of them, Uncalled and SIGMAP, because these are the ones that can be used as pre-basecalling filters without any changes. And these are the open source ones that we could use easily. And the methodology is to compare the labeling mechanisms of these approaches. So basically, these Uncalled and SIGMAP, they have to have a labeling mechanism that will predict if the raw signal is similar to any reference genome. So instead of doing this labeling for targeted sequencing, we just use their code to do this labeling for pre-basecalling filters, and then we compared these labeling mechanisms with TargetCall's labeling mechanism. And we have different use cases, but we have different evaluation metrics, and I will go through these evaluation metrics when I'm presenting the results. We have various read sets from various organisms, and some sampled from prior work, some simulated using state-of-the-art simulators, but we open source all of them so that anyone can reproduce our results. And we have different reference genomes with various reference genome sizes and ratio of useful reads. To evaluate TargetCall, we use NVIDIA 100 and Titan V GPUs for the light call component, and an AMD system with 0.2 terabyte main memory for the similarity check component. But we have a different system, slightly different system for SIGMAP and Uncalled with 1 terabyte main memory because they require more memory than we have in the first system for large reference genomes. So they actually don't scale well to the large reference genomes. And the first result we have is to show the benefits of pre-basecalling filtering in general. And we have different use cases in the x-axis. And our evaluation metric is the execution time of Bonito, which is the state-of-the-art base caller, on the entire read set, compared to the TargetCall's execution time for filtering all reads. And also the bonitos, like doing base calling on the reads that pass the filter. So it's actually end-to-end comparison between base calling and pre-base calling filtering and base calling useful reads. And we show that TargetCall can provide 3.3x base calling speed up over bonito on average. And next, we compare TargetCall against these prior approaches, SIGMAP and uncalled. Here we have 4 different labels in our x-axis because we have 2 different reference genomes labeled as CHM13 and HE38 for the sepsis use case. And now in the legend, we have SIGMAP uncalled and TargetCall, like prior ones. And we have a couple of different evaluation metrics. The first one is the execution time, where we compare the filtering time of these tools as well as the base calling time of the reads that pass the filters according to these different mechanisms. And our first observation is that TargetCall can provide up to 9.7x speed up over prior tools on average. And more importantly, it provides higher speed up improvement compared to prior ones when the reference genome size is larger. More specifically, it is 13.3x faster than SIGMAP for the large reference genome, and we couldn't even compare it against the uncalled because it's just inapplicable and it cannot generate the required indices for this reference genome. Next, we compared the recall of these tools, and recall refers to the ratio of useful reads that pass the filter among all the useful reads in the dataset. And our first observation is that TargetCall's recall is very high, which is more than 99%. And on average, it provides up to 23% better recall compared to prior tools. Similar to the performance improvement, recall benefits of TargetCall is also more important when the reference genome size is larger, where on average, TargetCall provides 15% better recall compared to prior tools. Lastly, we slightly changed one of the experiments to evaluate what would be the recall of finding human reads of these different tools. So basically, we have a sample, the same sample of the sepsis use case, and instead this time we try to identify the human reads instead of the bacterial ones. And the recall for finding human reads for SIGMAP and uncalled is between 40 to 54%, whereas TargetCall provided more than 96% recall for the same scenario, which leads us to the conclusion that TargetCall consistently provides high recall for all reference genome sizes tested. Finally, we compare the precision of these tools, and which refers to the ratio of useful reads that pass the filter among all the reads that pass the filter. And our first observation is that TargetCall's precision is more than 92%, which is significantly better than prior tools on average, 60% better than both tools, which shows that TargetCall can achieve better, the very high recall and precision at the same time, but these prior tools are optimized for high recall. That's why their precision is very, very low compared to TargetCall, which can provide good precision and recall at the same time. And we have more details in the paper about details of targeted sequencing, different light call designs. We have more details on our evaluation methodology, as well as many more exciting results, such as different light call architectures, the speed up, recall, and precision of different light call architectures we designed, and the accuracy and performance analysis of TargetCall across the entire genome analysis stack, and throughput comparison of light call against SIGMAP and uncalled, where we have more than 1100x better throughput, and peak memory comparison of these tools. Please check our paper for more detail. We also open source our code, scripts, and data sets in our GitHub page. And with that, let me conclude. We propose TargetCall, which is an accurate, scalable, and adaptable base calling filter that works in 2 steps. First, it does some noisy base calling using a lightweight base caller that would compute some noisy reads that are like very highly accurate but noisy compared to state-of-the-art base callers. And the second step is to compare these noise reads to a reference genome to determine if the read is going to be useful for the further downstream analysis. And our results show that we can significantly improve the base calling performance for all use cases we tested by filtering out majority of the user reads with high sensitivity in keeping useful reads. And also we can have better recall, precision, performance, and throughput than prior targeted sequencing approaches if we repurpose them as pre-base calling filters. I think that's all, and thanks a lot for attending. Do you have any questions?
Thank you, Banu. Questions? Okay, so yeah, so I'm gonna go first. So Banu, I was wondering if you considered comparing with squiggle filter and squiggle net. I mean, maybe not quantitatively, but at least qualitatively.
Yeah, we actually did that. So we couldn't compare against GoogleNet because this actually requires us to retrain their networks for all kind of different use cases we tested. That's why we said they cannot be applied as general purpose pre-based calling filters. And squiggle filter, we could actually compare it against it. Maybe not their hardware implementation, but their underlying dynamic time warping algorithm. But that would be a very actually slow method. Because it's a quadratic time algorithm, and it's like the quadratic time algorithms are even costly for sequences, and for raw signals it's even more costly because the length of raw signals are much longer than the sequences. That's why we didn't compare, because it's not a good baseline, it's not the state-of-the-art baseline, but we mentioned both tools in our paper and explain why we did not compare against them and their weaknesses in the paper.
Okay, makes sense. No, I was wondering, really, it's hard to compare quantitatively. Yeah, that's true.
Yeah.
Yeah, I have a question. So have you tried to extend TargetCall to, or maybe it already does, to handle multiple references?
Multiple references. It's just so what you can do is you can just append different references into a single file, and when you're running Memory-Map2, you can just give all the references as a reference, and you can just use it.
I see. And does the precision get worse if you have very large sequences?
No, actually, for example, in this viral use case, we have 10 different viral reference genomes in the target reference. So basically, we used it, and the precision doesn't go worse.
Okay. Thanks.
But yeah, I mean, we didn't extensively test it across like maybe 100,000 reference genomes, but I don't think so. As long as Memory-Map2's precision is good, it will have a good precision.
Anyone else?
Well, thank you very much, Banu. Great talk, great presentation, great work. Thank you.
Thank you.